We're going to move on to our next and final speaker from this session, Dr. Sunney Xie, who's going to tell us about high precision single-cell whole genome sequencing for PGD and PGS. Thank you.
Thank you. If I could have the slides, please. First, I'd like to thank Dr. Zhang for giving me the opportunity to speak here today. It is a real pleasure to attend this exciting symposium. So my group has been working on single-cell whole genome amplification. That is, if you give me a single cell, I would pretty much report to you the entire genome. At this point, there isn't a machine that could sequence these 46 chromosomes from the beginning to the end, so we have to rely on whole genome amplification. So I will tell you our latest method for WGA today, and then present two applications to PGD and PGS. The first one being how to avoid any mistakes in PGD, PGS in terms of sequencing. And the second being how to do this in a non-invasive way. The first whole genome amplification method attempted was PCR. It's been more than 30 years of PCR. PCR has a single copy sensitivity. But if you want to get the entire genome, it has a strong sequence-dependent bias. Some genes are amplified thousands, million times higher than others because PCR is exponential amplification. Roger Laskin, 14 years ago, invented MDA, multiple displacement amplification, which provided higher coverage, but it's still an exponential amplification. So here I illustrate, if I have a gain of 10,000, on the left side by exponential amplification, you see, assuming these two genes differ in amplification yield. One is 80%, the other is 100%. After this amplification, the counts, the intensity will be a factor of 24. If I do linear amplification on the right side, then I would still have this 80% difference. So in 2012, my group developed MALBAC, multiple annealing and looping-based amplification cycles, similar to the vine of MALBAC. We only amplified the genomic DNA linearly, and we protect the products such that by forming these hairpins so that they don't get amplified, we don't make copies on copies, and then at the end using PCR to bring the final product to microgram level subject for sequencing. So this allows us to obtain very accurate copy number variation measurement and avoid allele drop-off in a significant way, but it was still not quite good enough. So our latest method is based on transposon insertion. This insertion is random across the genome. Normally a transposon has a loop. We use this pre-cut transposon. This is a transpocyst dimer, and once it's inserted, then we have this two sequences that could use for amplification. Now, the key here is to do the completely linear amplification. We do that by in vitro transcription overnight, not too long. This gives us much higher, much better coverage, much lower allele drop-off. So here's the raw data comparing MDA, MALBAC, and DOPE PCR. You see MDA is noisy because this variation along the genome is not reproducible from cell to cell. Now, MALBAC, if you normalize, because the sequence dependence is reproducible along the genome. So once you normalize, it's better. But you see, LIANTI is at all scales. The vertical axis is the variation coefficient. That is the standard deviation divided by the mean. And of course, it's dependent on the noise is dependent on the bin size. But at all scales, LIANTI superseded. It's very flat, almost as flat as the bulk. But you see, previously we had megahertz resolution for MALBAC for CMV detection, just because of this variation. But now we can detect micro CMV. We have this so-called digital counting. So this gives us additional accuracy. Because we only have 2 alleles from a cell, you see the amplification products of these random fragments, they all have if they have the same starting and ending position, then we know it's they are from the same fragment, right? So we have this allele counting. Now here's an example of a 2-to-1 microdeletion with a kilo-base resolution. Here's a 2-to-3. And 2-to-1, imagine for DNA repair, these events are not synchronized inside the single cell, right? So if you want to know the double-strand break that causes microdeletion, this is a very accurate way of doing it. But today I will only talk about application to PGD, PGS. What about SMV? Now, I talk about this allele drop-off. If you don't amplify this allele, then this heterozygous mutation would be seen as a homozygous. So that's false negative. False positive can come from the amplification errors. Now, in the case of exponential amplification, if you have a mistake in the first cycle, then you would have it forever. You will propagate down. But if you do linear amplification, it's not so bad. Let's say the polymerase has a 10 to minus 5 error rate. Here's a comparison of the SMV false positive for LIANTI MDA and MALBAC and DOPE PCR. Even though we use a sort of a very poor polymerase, but the virtue of a linear amplification allow us to have a much lower false positive SMA detection rate. So let me quickly conclude this part of the talk. We have developed a new single-cell whole genome amplification method that relies on pure linear amplification with no random primers, but very random insertion of transposons. So LIANTI provides the highest genome coverage. We can easily get 99%. And it has good cell-to-cell reproducibility. It superseded MALBAC and MDA on CMV precision. Essentially free of sequence-dependent bias and rich kilobase spatial resolution for micro-CMA detection. And LIANTI superseded MALBAC and MDA on SMV precision. Both false positive and false negative rates are better. So using MALBAC at the end of 2013 in collaboration with Dr. Jie Qiu and Fuchou Tan, my colleague at Peking University. Dr. Qiu will be here this afternoon. We demonstrated that we could sequence the 2 polar bodies of embryo and use that to deduce the genome in the fertilized egg is normal or not, is viable for implantation or not. And that, of course, could simultaneously avoid the chromosome abnormality, which you all know is dependent on the age of a female. But it could also come from the sperm. And of course, if we sequence the 2 polar bodies, we would only see the problem from the oversight. But that has a higher probability. We could also deduce the point mutations from the mother. And I think you're all familiar with this. I just want to mention that this biopsy is all invasive. So no matter how good the WGA method is, let's say if I only have 1% false positive or false negative, in this business, you all know you cannot make any mistakes. I mean, Dr. Chell's clinic has 20,000 cases of IVF each year. So how do we avoid any mistakes? Now, people have been using lineage analysis. So we're not trying to detect a de novo point mutation. We're only trying to tell whether the allele that has the devastating point mutation is there or not. That's much easier, right? So traditionally, people do it with STR, genotyping, and more recently, the carrier mapping developed by Alan Handyside, Illumina commercialized it, worked very well. But these lineage analysis procedures are not quite compatible with the previous PGS, PGD. It's not compatible with next generation sequencing. So we published this paper the end of last year. We reported a single step next generation sequencing based PGD PGS procedure for linkage analysis. So basically, by using MALBAC to amplify the embryos, we could, this acronym it turned out to be the name of another Italian mine. So how does this work? So the key idea, we just use this as an example. This was the first patient who the man suffered from the hereditary multiple exostoses, all due to this one point mutation at EXT2 gene. He got that from his father got from his grandfather, his wife is fine, so it's a frame shift mutation. So we could look at the CMV, and you see the embryos with aneuploidy, and we can put a PCR primer, but all of this might have false positive and false negative. I mean, the SMV. This one we have the really high accuracy, and with LIANTI, I mean, you're not going to make mistakes on CMV. Now you don't even make mistakes on micro CMV. Okay, but the SMV is a problem. So we look at the SMVs around that point mutation. You know from sequencing the blood of his father, his grandfather. Right? So you look at the lineage analysis is you look at multiple SMVs of the same sample. Now the red one has the problem that okay, so we sequence the mother and father, the father's father. So the red one has the wrong allele. The point here is you look at these positions adjacent to this. It has to be adjacent because you could have a recombination, okay? But there are enough points, and they're consistent. So if let's one particular position, say here, for some reason you couldn't detect it or you detected wrong, you will be able to double check, and with 10 of them, you would have basically just eliminate the false positives. So of these 18 embryos, the red ones had undesired point mutation. The blue ones had aneuploidy. Dr. Chow selected number 4, and the baby was born. That was the first case. The second case, the problem came from the female's X-linked disease, and the symptom, some of you know this actor has been campaigning for research on this disease. So this is all due to one point mutation and EDA1 gene. And so in this case, we did the linkage analysis by sequencing the parents and their first son. Now, because it's X-linked disease, so this son, their first son, got the wrong X, the problematic X. Okay, so he suffered from brain damage only because he couldn't sweat when he was young. So this is the CMV, and we're missing embryo 5, and that was because there was a mechanical problem. So in the end, we were able to do the 2 polar bodies of embryo 5. So we also did the linkage analysis of the polar bodies. So again, the idea is just to look at multiple SME points. Okay, so of the 5 embryos, the red ones had the undesired point mutation. The blue one had the aneuploidy. Dr. Chow transferred number 2, and so the baby is healthy. We celebrated birthdays, and let me quickly summarize. MARSALA is a one-step NGS-based PGD PGS procedure with linkage analysis. It simultaneously avoids aneuploidy and point mutation that cause monogenic diseases, and significantly reduce the false positive, false negative of SMB. And it is cost-effective. We only sequence with 2X. Why is that? Now, I don't know if this will still be true for LIANTI, but MALBAC did have residual sequence dependence, so it amplifies certain regions reproducibly higher than other regions. So because of that, we could do relatively shallow sequencing 2X, so that's not too expensive. And this procedure works for autosome and X-linked diseases and male and female carriers. So this 2 cases was published in our PNAS paper end of last year. So how do we do this non-invasively? The biopsy, no matter it's at polar body stage or the blastocyst stage on the 5th day, 6th day, is still damaging. So there have been reports about the detection of DNA in the span culture. Now, this particular one from China was actually the closest to our work. They were able to use PCR to detect the genes. So basically, during the embryo development in the first few days, apoptosis occurs. Some cells basically has some DNA leaked out to the span culture. Now, we really need a high sensitivity, accurate analytical techniques to analyze this in order for this to be useful. So as far as we know, no previous studies have report validations of using this for non-invasive PGS. So this is done in collaboration with Shijia Lu former graduate student who was a co-inventor of MALBAC, went back to China, co-started econ genomics with me. This is my disclosure of potential conflict of interest. He collaborated with Dr. Cai in Wuxi and Dr. Yao in Nanjing. And Lei Huang is a postdoc in my group. We work together to do this. On day 3, we change the culture media and then collect the media day 5, day 6, and for sequencing. Now, we verify the result by sequencing the on the 5th day by doing blastocyst biopsy. That's the way everybody doing it. Okay, so we compare the result of NICS, that's the data from span culture, and the blastocyst. Now, in this embryo is consistent. This is consistent. This is not. So here I show you this table of 42 embryos. These are the consistent ones. 21 are consistent. This 15 are both abnormal. So these are normal. So we do have false positives and 2 false negatives. So with this, we could calculate the sensitivity, specificity, and that is the true positive over the positive and true negative over the negative, and then the precision, or sometimes called the positive predictive value, and then the negative predictive value. Now, none of them are 100%, but I would say this is pretty good considering we're not even touching the embryo. Okay, and so the question is, of course, this will improve, and I will tell you, I'll give you some clues about how to improve. But the question is this going to be useful? Okay, so the question I got a lot, and this is why Lei is here today. If you have questions, I unfortunately have a flight to catch after this. People ask just how much DNA is in the span culture. Well, how do you determine that? You can do MALBAC, you can do MDA, but how do you calibrate? So it's hard to. We determine the genome coverage for this embryo is 24%, this one is 33%, and this one is 65%. Now we know if we have a single cell, complete cell, that genome coverage will be about 70% for MALBAC. So we're low in that. So that suggests to us the DNA in the span culture is a fraction of a single cell. Now, if we have more than one cell, then this coverage would saturate to 1, right? So, okay. Then we zoom into this part, okay? So that was 24%. You zoom into this part, then you do see you're missing a lot of fragments. So even though we're missing, say, a good 70%, this CMV plot is still flat. So we're losing everywhere. We're losing them uniformly, okay? So that's still good. That would still allow us to determine whether the embryo has aneuploidy or not. So based on this, we estimate the total amount of DNA in each span culture is equivalent to that of a fraction of a single cell. Okay, so now you might ask, what is the origin of the false positive and false negative? The false positives, we believe, come from contaminations from the maternal cumulus cells. We had to do ICSI. it's important to do ICSI, then you only have the DNA from one sperm. Now, what about the false positives? We think they come from, actually, I apologize. I did this too quickly. The false positive comes from the mosaicism. So basically, you all know you could have cells that have aneuploidy generated, but they might not last very long, so they will undergo apoptosis. So now, when that happens, you could have abnormal copy number in the sperm culture. Okay. Now the question is, what is the implication of this false positive and false negatives? Now, if all the embryos that you look at it are false positives by NICS, then you could still, on the 6th day, because you have no good ones, you might as well just do the invasive biopsy on the blastocyst, right? So that's not too bad. Now, what if it's a false negative? Well, usually they do not result in successful pregnancy. So overall, the existing false positive and false negative is not that bad. Okay, so the first patient Dr. Xie has 3 embryos. Embryo 1, embryo 2 had aneuploidy seen from NICS. He immediately checked on the blastocyst. They are consistent. And the 3rd embryo by NICS is normal, so he didn't check. He didn't want to touch it, right? And the baby was born. That was the first baby here. So of the 7 patients, Dr. Xie and Dr. Gao have done, 5 of them have achieved live birth. 1 is still waiting, and 1, the implantation failed. But 1 out of 7, so we believe we are seeing the improvement by this non-invasive method. But of course, this will we have to wait until a larger clinical trial. So in conclusion, the NICS method avoids the need for embryo biopsy, and by using MALBAC, actually LIANTI as well, we can perform the next generation sequencing on the sperm culture media. And we have characterized the precision, and I think the precision is good enough, and it's actually quite good considering we're not even touching it. So 6 of them have achieved successful pregnancy, and 1 more need to be born. And let me finally thank the people who did MARSALA, Dr. Qiao, her assistant, Dr. Yan, in collaboration with Shijia Lu and Lei Huang. And these are the people responsible for NICS. Of course, it's a large team, postdocs in my research group at Harvard who developed LIANTI. We got a lot of help from Dr. Heng Li at Broad Institute. And I want to thank you for your attention.